Hello, I'm Christine Curcio, and I thank you for listening today. This is part 3 of a 4-tutorial series on deposit-driven AMD. Here we will focus on subretinal drusenoid deposits, or SDD. The original name was reticular pseudodrusen. Here are my supports and disclosures. Here are the key takeaways from the 4-part series: that AMD is a solvable 4-dimensional puzzle visible in vivo with OCT-anchored multimodal imaging. The photoreceptors and the RPE together are a sensory chip of the brain with the geometry of the foveal singularity. We discussed the value of a 3-layer versus 5-layer Brooks membrane. The essential AMD pathology is extracellular deposits on either side of the RPE. Drusen follow the cone vision system, whereas subretinal drusenoid deposit, which we will discuss today, begins where the rods are numerous. And what we learned from lipid compositional studies is that the dietary input to the outer segments may be a defining feature. To review, the OCT-B scan on the left shows the layers affected by AMD. The schematic shows a progression from normal aging on the left to advanced AMD on the right. The bands arise due to the precise horizontal alignment of millions of vertically compartmentalized cells. There are multiple biologically distinct layers within 100 micrometers of vertical height between the ELM, the dashed line on the top, and the choriocapillaris endothelium below. Here are the inner segments of the photoreceptors, the outer segments, the interdigitation zone of the outer segments and apical processes together, the RPE cell bodies, and Brooks membrane choriocapillaris together. These bands may be visible, perturbed, or absent in an AMD eye. Layers are added representing extracellular deposits. SDD between the photoreceptors and RPE, the subject of this tutorial. Basal laminar deposit between the RPE and its natal basal lamina, and drusen between the RPE basal lamina and the inner collagenous layer of Brooks. Let's begin with some history about SDD. Why are there two names? Because they started from different visualizations. And an important point is that SDD were omitted or mixed with drusen in fundus grading systems for AMD. Over the years, this patterned biomicroscopic sign, shown here in color, infrared reflectance, autofluorescence, and OCT, has been called by many names depending on the investigators and the detection technology used. The names that stuck are reticular pseudodrusen from the ENFAS view and SDD from the OCT view. The story started in 1988 when Shirley and John Sarks illustrated but did not name these deposits in their electron microscopic study of geographic atrophy. You can see from the quote, the photoreceptor outer segments terminate in focal collections of membranous debris, which are partly surrounded by the inward extensions of the RPE. Membranous debris was the Sarks name for the main ultrastructural component of soft drusen, thus implying that both deposits were the same. Specific techniques applied over the years showed that they were different. In 1990, Mimoun et al. In France described drusen visible in blue light. This slide shows the blue channel of a digital color fundus camera, and the pattern of dots is apparent. The first case of reticular pseudodrusen to come to histology was missing a retina. The most notable thing about the specimen was the thin and fibrotic choroid, which turned out to have a clinical equivalent much later. Direct clinical pathologic correlation of an eye with a reticular pseudodrusen and a retina in place, also by the Sarks, definitively place these in the subretinal space. Regular spacing accounted for the patterning. I prefer the name SDD because the deposits are not always reticular. They are real and not pseudo, and they are distinct from drusen. However, SDD is the name used for histology in OCT, and RPD is used for the fundus view. Here are the three stages as described for OCT. They disturb the overlying ellipsoid zone, EZ, of the photoreceptors. And here is histology showing the extracellular deposit anterior to the RPE in an intact retina. We knew at the time that this was not an AMD eye. SDD were unappreciated until recently because they appear late in life in a technology-dependent manner. OCT on the bottom is the clearest, but the pattern is also visible in near-infrared reflectance, fundus autofluorescence, and color. In the large population-based Rotterdam study, the combination of color and near-infrared reflectance shows best the high prevalence of SDD in the oldest age group. So color fundus photography is widely used, but it's relatively insensitive. And the grading systems thus combined drusen and SDD, adding noise to the risk estimates for both. Now we will look at some of the studies that establish the separate identity of this deposit. In their description of membranous debris, the Sarks-Killingsworth group pointed out this material in three extracellular locations in AMD eyes: in drusen, in basal mounds inside the basal laminar deposit, and in the subretinal space. They speculated a photoreceptor origin for the latter. Now, a general problem is that in postmortem eyes, the retina frequently detaches. It is possible that this deposit is just damaged cells. So we were fortunate that in AMD eyes with attached retina, we could use Philippine histochemistry to show that, for unesterified cholesterol, to show that both drusen and SDD were 3 times brighter than the outer segments. This is striking due to the high concentration of membranes in outer segments, shown here. So one thing that we got right in that early paper was that the high intensity of Philippine fluorescence for unesterified cholesterol indicates that a pass-through of photoreceptor outer segment membranes cannot account for the quantity of cholesterol without a specific enrichment mechanism, additional sources of cholesterol, or both. But one thing we got wrong in that early paper was the idea that apical extrusion of unesterified cholesterol-rich material represents deranged polarity of a normally vectorial process for basolateral lipoprotein secretion. Separating SDD from drusen was still in the future. Here are the 3 stages of SDD in spectral domain OCT. And here is clearly the extracellular material between the RPE and the photoreceptors, and you can see the extensions of the apical processes in bundles punctuating the deposit. Here, an isolated deposit has on its top a cap of material that looks like it might have derived directly from outer segments because discs are still visible. Clinical imaging has revealed clearly this relationship of deposits to the rods and the cones. On-FOS OCT of these 3 patients shows that the type of deposit depends on the local rod-cone ratio, which is shown in color in below. Drusen form under the fovea, where cones are numerous, and SDD in the solitary dot morphology, form where rod-cone ratio is low, and SDD in the ribbon morphology, form where the rod-cone ratio is high. Because of the differential composition of drusen for forms of cholesterol by histochemistry, We postulated that SDD and soft drusen were two parts of one system of lipid transfers to serve specific aspects of cone and rod physiology. You can see that SDD is rich in unesterified cholesterol, whereas drusen has that plus esterified, so there was really a major difference in the lipid content. We could further speculate that this differential membrane structure and maintenance mechanisms of the outer segment of rods and cones might be the basis of those differences in lipid transfers. Robin Geimer and Erica Fletcher published superlative histology of a surgically removed eye that was previously imaged by OCT. They showed the correspondence of type 3 SDD with the large solitary deposits. The deposits were strongly positive for vitronectin, and the largest ones could appear poking into the outer nuclear layer. The Mueller glia responded to even tiny SDD by upregulating GFAP. Microglia invaded from inner retina, and cone opsin was apparent in SDD, and they were oil red O negative, confirming our results. Finally, in flat mounts, the RPE could be seen to be perturbed under the deposits. In 2013, Rick Spade demonstrated that SDD are dynamic. SDD present at baseline on the top disappear from superior macula 3.5 years later on the bottom. This is called outer retinal atrophy, in which the photoreceptor layers simply collapse. Over the same time, the deposits appear in inferior retina. In this histologic example of outer retinal atrophy, photoreceptors shorten where the SDD have disappeared. The RPE is intact, basal laminar deposit is continuous, and the Mueller glia become gliotic. In this longitudinal imaging study using adaptive optics and magnified OCT through the same deposits, individual SDD can be seen to appear, grow, shrink, and morph. SDD are associated with risk for type 3 or intraretinal neovascularization. Here is an eye with type 3. You can see the deposits, and in the fellow eye, there's some atrophy, and the deposits are clear, and here they are in histology between the RPE and the photoreceptors. Up to 1/3 of presenting neovascularization is intraretinal in origin. It tends to be bilateral. Over 50% of the fellow eyes progress within 3 years. There's poor visual prognosis due to macular atrophy. This form of neovascularization is very responsive to anti-VEGF, but it's difficult to diagnose, and the precursors are still being identified, and SDD has an important role as a precursor. To investigate the AMD deposits, we created a website of AMD pathology called Project Macula with 132 postmortem eyes collected over 14 years. They were processed for high-resolution histology and electron microscopy preparation viewed by light microscopy. Sections through the fovea and superior perifovea were systematically reviewed and photodocumented. Thicknesses of chorioretinal layers and contents of the subretinal and sub-RPE spaces were measured and annotated at predefined locations shown below. The histology, sampling scheme, and photomicroscopy was designed under the hypothesis that the photoreceptors drove the metabolic demands on the support system and that the signal sources for OCT are subcellular. Our analysis showed that SDD abundance is low, but not absence from the fovea, and they're high in the perifovea. This distribution differs from that of drusen and basilinear deposit. The difference between AMD and control eyes is larger than it is for drusen, which might be real or artifactual due to detachments. Wu et al. Reported SDD locations in the zones of the ETDRS grid in 137 AMD eyes. SDD are most abundant superior to the fovea in the outer ring of the grid. This corresponds to the rod density hotspot in the retina. Interestingly, type 3 neovascularization does not occur where the deposits are. Haj Najib et al. Mapped type 3 in 78 eyes and found that 92% of them occurred in the inner ring of the ETDRS grid. We found a similar distribution of 7 vascular lesions in a pair of eyes with type 3 neovascularization. We can look at this one right here in the next image, and you can see the neovascular stalk connecting the deep capillary plexus to the top of calcified drus. So our current thinking is that SDD indicates an eye at risk, not specific cells at risk, because of this topographic non-correspondence. Because of the overall hypothesis that deposits reflect specific physiologic needs of cones and rods, it is justifiable and helpful to directly compare SDD and high-risk soft drusen material. In this tutorial, we discuss the difference in retinal distribution, lipid content, and the predilection for retinal versus choroidal neovascularization. Equally cogent differences are that drusen progress to hydroxyapatite or calcification, but SDD do not. Also, drusen are positive for carbohydrate stains, and SDD are not. Protein composition is partially overlapping, with CD59 clearly different. Prophylactic laser reduces progression risk from drusen but makes it worse for SDD, and the responding cells differ. SDD occur in several diseases involving Bruch's membrane or transport, particularly of retinoids, and soft drusen are specific to AMD. Now I will present new data on differences in ultrastructure and evidence for lysolipids in SDD. By electron microscopy, the electron-dense lipid-containing material in SDD are whorls that differ in size and morphology from the lipoprotein particles in soft drusen on the right. Now, lipids are essential for fuel transport membrane signaling, and they're defined as fatty acids and derivatives, that is, a hydrocarbon change and a terminal carboxyl group, often complexed with head groups. The phospholipids are the main component of lipid bilayer membranes, and hydrolysis here results in a lysolipid and a fatty acid. In the next slides, the formula that I will show indicates the number of carbons and the number of double bonds. M over Z means mass over charge or molecular weight. Let me introduce imaging mass spectrometry, a powerful labor-free method that produces images based on molecular weight of tissue components without antibodies or other stains. It's perfect for layers of retina seen here by ex vivo OCT. Here is a tissue section sprayed with a matrix, ionized by laser shots on a geometric grid. Ions are captured and maps of molecular weight are generated. By capturing microscopy images of tissue before and after ionization, the weights are localized precisely. The resolution is set by the grid spacing of 10 to 15 micrometers. With accurate mask from IMS, lipids can be identified by way of database search. Also, very tiny samples can be captured for direct assay. The top is a clinic patient with SDD, shown at the arrowheads. The bottom shows an eye of a different eye donor with ex vivo OCT, which is how we can find OCT for IMS studies. Here are representative lipids from positive ion mode, including lyso-PC, 2 sphingomyelins, and a phosphatidylcholine. Column A shows IMS signal plus histology. Column B shows IMS signal plus tissue autofluorescence, and column C magnifies A for detail. The important point is that each lipid has a specific location. This sphingomyelin is exclusively found in SDD. This PC in SDD is also in adjacent photoreceptors and in chorea capillaris. This sphingomyelin notably avoids SDD but appears in the uninvolved RPE. And this lyso-PC is in SDD and chorea capillaris. And these are taken from over 900 positive ion mode signals, and we had almost as many for negative ion mode, which could be used for automated analysis. And the top 5 hits, which I'm just skipping over for now, were all lysolipids. Now, lysolipids are actively studied in many organ systems. They are generated by the activity of lipases on cellular membranes, shown at the left. There are 50 phospholipase A2 enzymes in 4 families, and secreted PLA2 act extracellularly to hydrolyze membrane phospholipids. The long-chain fatty acids released by PLA2 give rise to bioactive lipid mediators with pro- and anti-inflammatory action, depending on the organ system or disease. Docosahexaenoic acid, DHA, is an omega-3 fatty acid, and it's essential for cognition, development of brain and outer segments, and formation of the blood-brain and blood-retina barriers. DHA is transported in plasma as a lysophosphatidylcholine, and the lysolipid receptor MFSD2A on the RPE takes up DHA for outer segment membranes. In 2011, authors from Moorfields Eye Hospital announced that benign fleck retina, an inherited retinopathy first described in 1980, could be linked to mutations in the PLA2G5 gene, which encodes a group 5 secreted phospholipase. Here is a fundus of an affected patient, and here is an SD-OCT of the flecks. 5 years later, these authors described this as an SDD phenotype. This disorder is very rare, but the findings have been replicated by several groups. We thus considered PLA2G5 a candidate lipase to be involved in SDD formation. This immunohistochemistry of 2 donor eyes shows brown reaction product. In the photoreceptor inner segments, cones more than rods, and in the RPE. To summarize the significance, we are familiar with the convergence of genetics and deposit composition with regard to the role of complement in drusen, leading to developments ultimately resulting in approval of new complement inhibitors for AMD. Here's a convergence of a different type. The monogenic inherited retinopathies like Sorsby, fundus dystrophy, and Malatia leventinisi have phenotypic overlap with AMD. providing a single gene that can be exploited experimentally to create new cell and animal models. We can hope that similar progress can be made after finding the convergence of lysolipids in SDD and PLA2G5 expression. A limitation of the study that I just showed you is the small sample number. More data are definitely needed about SDD. To wrap up about SDD, these start where the rods are numerous. They are risk indicators and not precursors for type 3 intraretinal neovascularization. They appear first near the hot spot of high rod density. They appeared in older patients than those with drusen. That a major component is lysolipids, possibly indicating outer segment remodeling. They are ultra-structurally and molecularly distinct from the cells around them and from other. Here are the key takeaways from the 4 tutorial series on deposit-driven AMD. Thank you for your attention today.